cosmic storm. Floating on whimsical wavelengths is the norm. Dancing through the stars, chasing spectrums of light. Riding the waves that are just out of sight.
So one of the things that I've noticed is that I don't introduce myself very often. And for all those new listeners out there that show up from time to time, my name's Dr. Jeffrey Zurich. I'm the host of Whimsical Wavelengths. And today is all about the white sturgeon. Because I've spent the vast majority of my life living along the major rivers in southern British Columbia, Canada. The Kootenay and the Columbia Rivers, and now the Fraser River. These rivers have a lot in common from the ecology standpoint with respect to salmon and the subject of today's episode, the white sturgeon. And these fish are amazing. I've spent a lot of time fishing the Columbia for trout and walleye, also known as pickerel in other parts of Canada, but I've never interacted with a sturgeon. To me, they've always been mysterious fish of the river. I know they are there, but I've never seen them. The Loch Ness of the Columbia and the Fraser River. And I've always wanted to learn more about these creatures. These group, sturgeon, and their family group, they first appeared in the late Cretaceous, with the oldest seen in the fossil record at 100 million years ago, found in Alberta, Canada. In fact, they look today very similar to what they looked in the late Cretaceous. They join a short list of creatures that have not, at least outwardly, significantly changed their body shapes or their large scale building blocks in a very long time. Like the crocodile, whose modern body shape came together 80 to 90 million years ago, or the horseshoe crab, which has been around a lot longer, nearly 400 million years. Besides being what some term a living dinosaur, they're also polyploidy, meaning they can have different numbers of chromosomes. This all part of it actually makes my head hurt. Give me the simplicity of a wave equation. Thankfully, in a few minutes, I have a guest to help me understand it. But let me attempt to set this up. So the white sturgeon, are scientifically amazing already, and we haven't even gotten into what the species do, above and beyond what the local rivers mean to us. They're a culturally important food for First Nations of North America and harvested for caviar in other places. The caviar can go for $1,000 to $5,000 Canadian per pound, a true luxury item that starts its history almost 2,500 years ago, when the Persians ate them in a belief that it provided strength, later becoming a prized delicacy of European nobility. Sturgeon are predators and scavengers using barbels or whisker-like sensory organs that smell and taste to detect on the riverbed things like salmon carcasses, eggs, lamprey, crayfish, clams, and worms. Some might call them a keystone scavenger, aiding in keeping riverbeds healthy for other species. Now we have the geologic time perspective and the culture perspective and how they are important to the river systems they inhabit. What about their population? I can anecdotally attest to these fish being rare. In fact, they're threatened as a species due to overfishing and habitat modification. The Columbia River has many, many dams along it due to the Columbia River Treaty. All right, time to bring in the guest. To help us understand about these enigmatic fish, their populations, and their genetics, please welcome a fisheries geneticist with the Hagerman Genetics Lab, Dr. Stuart Willis.
Hi, Jeff. Always excited to talk about sturgeon.
Yay! Before jumping into the science, I always think it's important to see how someone comes by their chosen subject. I mean, careers in STEM, we need to make them less scary in general, and science is always a human endeavor. I grew up living beside a river, but what about you? Was there a river, a body of water? Was it fishing? What brought you to studying fish and their ecosystems?
Yeah, so I did a lot of fishing with my dad as a kid back in Texas, except I always wanted to bring our catch home and keep them in an aquarium to learn more about them. So as a teenager, I did keep a lot of aquaria, but mostly South American river fishes, not our North American native fishes. And so then when I went to college, I got hooked up with a professor who had a graduate student doing phylogenetic and evolutionary ecology work on South American fishes, and I was hooked. And so I did my graduate work on phylogenetics and population genetics of South American fishes. But in academia, as you know, you sort of have to go where the money is. And so that eventually led into the work that I do today for a consortium of tribal nations for whom fish, and particularly salmon, are an integral part of their culture. They've taken a lead role in managing the natural resources within their ancestral territories in the Columbia River Basin. And as a part of that work, I manage our genetic investigations of white sturgeon, which are the largest freshwater fish in North America.
Okay. So, I mean, I grew up next to the Columbia. I fully support this sort of research and keeping those ecosystems at least understandable, what's happening to them, monitor them, and make sure they're there for the next generation. But are you also looking at different opportunities to do other research outside of those drainage catchments as well? I mean, yes, you follow the money, but I also want to go back one day and do some volcano research that may take years and years and years and years to actually come to fruition. I mean, that's the nature of sometimes playing science. It's a long game.
Yeah, I still do collaborate on occasion with people from my past. I still have a project that I'm working on looking at photosensory evolution in shallow and deep dwelling reef fishes and things like that. But for the most part, I stay within what the tribes need, and the tribes are focused on fisheries in the Columbia River Basin. A lot of what I do day to day is salmon genomics, and there's a huge community of people, as you can imagine, that are interested in all things salmon, everything we find out about salmon. White sturgeon is a little bit smaller of a community, but to me, because there's so much less known about white sturgeon, it sort of feels more like a voyage of discovery, much more like the things that I did as a graduate student, where I was going out into the Amazon and finding things for the first time, seeing places that no scientist had collected fish before, at least. So a lot of new stuff. Hard, difficult, but new.
Right. And we'll get into how you managed to find these fish, hopefully, eventually here. But I want to pivot a little bit to the rivers themselves that are kind of the focus of this study and your work in general. The mighty Fraser and the Columbia, two rivers here on the west coast of North America. I really felt like I needed to add a little bit Canadian content from the political past. It's kind of a fun little tidbit. The two rivers are separated by only about 45 meters of elevation. Like there's a divide right near Valemount, British Columbia. In the 1960s, British Columbia actually made a public threat in taking the Columbia's water in a negotiating tactic for the Columbia Basin River Treaty. In a geographic sense, the rivers are very similar, yet today are very different. Perhaps let's start there. What is kind of like the state of the two rivers?
Yeah. So both the Fraser and Columbia basins face increasing pressure from urbanization and other intentional and unintentional modifications, as well as changing precipitation and temperature patterns that make life as an aquatic organism exponentially harder. The Fraser, fortunately, doesn't have the same level of damming that Columbia does, particularly the main channels, banks, and going back to your comment on Canadian politics, as I understand it, to push back from the public about the effects that would have on aquatic species and the people that depend on them. Which is not to say that Fraser is in pristine shape. Channel modification and pollution are a big issue, and anadromous fish runs have been on the decline for a number of years, though some of the factors for that are local and some of them probably regional. The Columbia, on the other hand, is strongly segmented by hydroelectric facilities from the tributaries through the main channel. And on top of that, as well as urbanization and other habitat degradation, not to mention climate change, invasive species have really started taking their toll. Some intentionally introduced by well-meaning but short-sighted state agencies like channel catfish and shad. Some introduced by well-meaning but ignorant bucket biologists like walleye. And some completely unintentional hitchhikers like quagga mussels and Siberian prawns. So to answer your question, the state of the rivers is probably middling to struggling, respectively.
Right, but maybe where I was kind of wondering is like what's the effect of those dams? Because that's like all things being equal, they have. I mean, yeah, I could say the Columbia, at least in its lower reaches, has more population pressure, but they're very similar in their geographic and everything else, but the Fraser's dirty. It has a lot of sediment. I wouldn't say dirty. Dirty's the wrong word necessarily, because it's not like it's not like it's polluted or anything, but you have all these dams in the Columbia that are causing all that sediment to settle out. And that's, I've heard it said that a dam gives a river a stroke. You can partially recover from that stroke, but you're never going to get all the way back.
Right. So there are great diversity of dams on the Columbia. Sort of the two main types are run-of-river dams that sort of don't have a big difference in elevation, and then dams that have a large body of water that are held behind them. And those in particular have major effects on the river ecosystems because not only do they slow down the water, which means that fish now have to swim rather than just being in the current, but they also allow the water to heat. And that hot water is really hard for these very cold specific species, cold adapted species, because they get in hot water, their metabolisms ramp up, their parasite load is more difficult to manage. And so they're very different, both at a very small scale endocrine level and then at a major ecological level. And then, of course, there are a number of dams that don't even have fish passage at all. So it's hard to be an anadromous fish when you can't even get over the dam in the first place.
Okay, I was going to move on to the next question, but I need to check the word anadromous.
So anadromous is a term that just means fish that start out in freshwater, go and live part of their life in saltwater, and then come back to spawn in saltwater. It's a subset of what we call diadromous fish, so fish that have sort of two different parts to their life history strategy or life cycle. The alternative is catadromous fish that live in saltwater as adults, spawn. Their young go into freshwater for some period of time and then return back to saltwater as spawn. So I think that the American eel is a catadromous fish. They all spawn out in the ocean. Their young spend most of their life in freshwater.
So then what would salmon be?
Salmon are anadromous fish. So they spawn in freshwater. The young go out to the ocean, grow up, and then come back to adults. And this is sort of getting off on a tangent. There are some salmon that don't, right? Like rainbow trout. Anything we call a trout is because they can complete their life cycle in fresh water. The reason that they go to the ocean is because there's so much more nutrients in the ocean. So there's an advantage to going out to the ocean to be able to grow faster or have greater access to nutrients. And so all of that life history differences are adaptations that these fish sort of manage across time as environments changed.
Right. Okay, well, to pivot to the topic of the day, the white sturgeon. Who are they and what role do they fill in the ecosystem of these two rivers?
Yeah, sturgeon are very charismatic fishes for those who are lucky enough to live where they do. As you mentioned in your intro, they look very prehistoric. And indeed, as a group, the order Acipenseriforms, which includes sturgeons and paddlefish, had been around for a long time, being one of the earliest groups of bony fishes to break off at least 300 million years ago, so before the divergence of the dinosaurs from other reptiles. But I personally don't like the term living fossil or living dinosaur that sometimes gets applied because they are, in fact, highly modified both from other fishes and from their own ancestors. For example, they have a mostly cartilaginous skeleton, like sharks, but that's a feature that they evolved after diverging from other fish as a secondary characteristic. Similarly, while fish recognizable as acipenseriforms have existed since at least the early Jurassic, modern sturgeon and paddlefish were as different from those forms as sturgeon and paddlefish are from each other today. But, as you say, they have been around for a long time, and they have some neat features beyond their unique appearance. For example, they still retain electrosensory organs on their head called ampullae of Lorenzini that allow them to sense the weak electric field emitted by other aquatic organisms, a feature that they share with sharks and rays and one of the group of early diverging bony fishes. And you can imagine that feature helps them to find fish in the deepest, darkest parts of large rivers where they're found. And that leads into their ecological roles, which change depending on their life stage. As eggs, larvae, and fry, they are food for a lot of other creatures. But they grow really fast in the first couple of years of life, usually reaching 30 to 40 centimeters in the first year and a meter within 3 to 5 years. And after that, especially as adults that reach 3 or 4 or even more meters long, they don't have a lot of natural predators. So they can sit at the top of the aquatic food chain, eating basically anything they can hoover up into their mouth that smells good, which could be live or dead. And so they are both top predators and scavengers.
All right. So I guess they're kind of like a keystone species to some extent for these two rivers.
Um, it's hard to say keystone species because that sort of depends on your perspective. I mean, you could say that salmon are keystone species for these ecosystems because um, they bring all of those marine derived nutrients. They're an apex species, right? They sit at the top of the food chain, and certainly they have effects on the ecosystem. But keystone is sort of an amorphous concept, so I'd be cautious to venture out and say that for sure.
Well, how long does it take for them to get to adulthood? Because I know that they're long living, and generally speaking, those species who are long living, they take a long time to reach adulthood. They take a long time to reproduce. Is that the same for this species as well?
It is. So, as a survival strategy to avoid being prey yourself, white sturgeon grow as fast as they can to get into a size after, so that they're less likely to be eaten. But once they do, they slow down their growth and just sort of chill. Since prehistorically, at least, mortality among subadults and adults was really low, while conversely, finding food to maintain such large bodies is a full-time job. So they are a type of fish that exhibit what we call a periodic life history strategy, which means that they reproduce at regular intervals, usually in the late spring when snow melt is high, but the water is warming. But the local conditions that help their young to survive can be unpredictable spatially. And so it's really advantageous to produce a ton of eggs that get spread out across space to mitigate risk. And for organisms like that, for which egg number or fecundity is a determining factor in reproductive success, size at reproduction, which generally also means age, also becomes a determining factor, which explains why sturgeon males, for whom size is slightly less of a constraining factor, don't mature till around 12 or to 20 years of age, while sturgeon females don't usually start to mature around at least 15, usually 18, but can take up to 30 years to mature. And then once they do, because that kind of pulse reproductive effort is so taxing, females in the wild will usually only reproduce every 3 to 6 years or so. And some of those years will be good recruitment years, and some will be bad, and based on prevailing environmental conditions. And so the reason why they succeed is because they can live so long enough for a few fry to survive from a few of the 70, even 100 years that they're going to live. But that's in natural conditions and rivers unaltered by dams and dredging and pollution, invasive species, et cetera, et cetera. So how that continues to function in our modern river ecosystems is something we are actively studying.
I have a feeling I'm about to jump the shark here with this question because you kind of brought it up, but they take decades to, like, humans have a couple of years and we, sexually mature. Like it's a standard window. It's not exactly the same between individuals, but it's pretty close. Like it's not decades. And you just gave a range that were potentially decades long that it takes to mature. Like that seems like, could you imagine being a teenager for decades? Like and one fish goes through that in 12 years. The next one takes 20 years. Like, I mean, we make jokes about us men taking a lot longer to mature, but this is taking it to the whole next level, right?
It is. Yeah. And so some of that is probably environmental conditions. If you grow quickly, you're more likely to mature quickly. If your growth rate is sort of set at an early age to be slow, you're going to take longer to reproduce. But sometimes that can be an advantage. When we talk about, keep bringing it back to salmon, because that's one of the other things that I work on. Salmon have very distinct peaks in age at maturity, but larger fish have larger fecundities. They produce more eggs. And so it can actually be an advantage to become sexually mature later. And so that it's entirely possible that that's true for sturgeon as well, that it can be an advantage to wait to become sexually mature because you're larger. You end up having a greater fecundity. Maybe you weathered out a time period over which you wouldn't have been successful reproducing anyway because there were cruddy conditions for offspring. And so Yeah, that range certainly has implications for population dynamics, but at an individual level can actually be an advantage. And not to get too hard to jump too far ahead into talking about sort of state of the population. One of the things that we're seeing in looking at some of these river segments, the ones that are sectioned off by dams and that kind of thing, is that fish are taking longer to mature. And so basically, that's probably because these ecosystems are not as productive, or the fish don't have access to the areas where they historically would have been able to get enough food to be able to reproduce at younger ages.
So let's pivot towards the actual paper, the reason I got you onto the show. And to be honest, I really struggled with it to understand it. I mean, I guess I'm a volcanologist, this to be expected, but even the more I learned, the more I was blown away. The paper, Genome Assembly and Diagnostic DNA Markers for Sex of the Largest Freshwater Fish in North America, the White Sturgeon, Acinpenser transmountainus. I know I did a terrible job of pronouncing that, but that's okay. So I guess let's start with the hardest thing that I had wrapping my head around, and I made a joke, give me wave equations instead. What exactly is ploidy?
Yeah. So this paper is our first attempt at reconstructing the genome of white sturgeon so that we could develop new genomic tools for their conservation. But we really couldn't do that without confronting the issue of ploidy. And so hopefully I can explain that without getting too much into the weeds, and feel free to jump in if I'm getting entirely too nerdy and esoteric. But
Oh, we fully embrace the nerd here. Don't worry about that. Esoteric, well, to a point, because we have to understand it too.
well, that's my wife's euphemistic term for me. So
She calls you
that's just how I
esoteric.
But ploidy refers to the number of complete sets of chromosomes in a cell, whatever complete is for that species. And I'm using air quotes when I say complete, and my voice does not convey that. And I'll come back to that in a moment. But to give you an example you're familiar with, humans, like most multicellular organisms, are diploid. Di meaning two. That is, we get one complete set of chromosomes from mom and one from dad. So the final set contains pairs of each unique or homologous chromosome, meaning what each mom and dad contributes, eggs and sperm, are, in this case, monoploid. Mono meaning one complete set. Now, technically, we use the term haploid to refer to the complement of chromosomes in a gamete, an egg or a sperm. Hap meaning half. And what the combined gametes produce, somatic cells, are called euploid, the root eu meaning true or even. And then we call instances where there are incomplete sets of chromosomes aneuploid, or not even. For example, in humans, we may be familiar with trisomy, where there are 3 copies of chromosome. And the trisomy, 3 bodies of chromatin or chromosome, which creates uneven sets of chromosome or aneuploidy. And this is usually a problem developmentally, like trisomy of chromosome 21 in humans produces Down syndrome, because gene regulation, as it turns out, is frequently dosage dependent, meaning the regulation of expression of genes has evolved to be balanced in a copy number dependent manner. And aneuploidy messes up the typical dosage. But going back to the air quotes, complete set of chromosomes, when we look across animal and plant species, we see wildly different numbers of chromosomes and genome sizes, okay? Some of these differences are because chromosomes have expanded or contracted individually, or some chromosomes have split or combined. And so we see relatively moderate changes in chromosome numbers among related species. But those changes don't affect the ploidy, because it's not changing how many sets of chromosomes are in each cell, rather just the numbers that make up each set. But sometimes the genome sizes changes because of polyploidization, poly meaning many, in this case, many sets, where the number of chromosomes goes up by entire sets all at once. And there are 2 main ways this can happen. In allopolyploidization, allo meaning different, in this case, different species, 2 moderately divergent species hybridize. And through some cell developmental magic, the progeny end up with twice the combined set of chromosomes from both parental species. And this form of polyploidy is fairly rare in animals, though there's at least one group of fishes, the Catastomidae, or suckers, that are thought to be allopolyploids. But it's actually fairly common in plants, including some you might know, like wheat. Wheat is an allopolyploid species. If, on the other hand, polyploidization happens without hybridization, by the doubling of a genome within a species, we call that autopolyploidization. Auto meaning self. And autopolyploidization has happened repeatedly, not only in plants, but also in vertebrates, especially in fishes and in particular in sturgeons. So based on mapping of chromosome counts across the phylogenetic tree of fishes, that is the relationships among species of fish, we can estimate that the ancestor of the acipenseriforms, that was sturgeons and paddlefish, had a euploid or complete number of chromosomes around 60. But all modern acipenseriforms have at least 120 chromosomes in their genome. And many sturgeons, like the white sturgeon, normally have at least 240 chromosomes in their cells. So approximately double to quadruple the ancestral number. So if we presume that that ancestor with 60 chromosomes was diploid, which is a reasonably safe bet for a reason we can talk about, then the species with around 120 chromosomes would be evolutionary or ancestral tetraploids. That is relative 4 complete sets to that ancestor that had 2. And species with 240 or so chromosomes would be ancestral octoploids with 8 complete sets. And so the haploid number for those organisms, what's found in an egg or a sperm, and what's half the euploid number in all of those, is multiple complete sets relative to the ancestor. And big jumps like that, from 60 to 120 to 240 as the euploid number, are the hallmark of polyploidization.
Okay. To be honest, I had a hard time following. I think it's just because it's like there's not enough numbers. No. And I also couldn't spell any of those words. Like, I just come back to like the human idea here, and we talk about human genetics when we end up with, like, it seems like the more you have, the more things that can go wrong, or is it the more you have, the more fail safes you have? Like,
yeah.
Like, how does that work?
So it's what's really interesting is that in most of the ancestral polyploids we know, in both plants and animals, the genome actually acts as functional diploids, meaning the same 2 pairs of chromosomes pair up and split for distribution of gametes rather than in sets of multiples or in random pairs of the original multiples. We call this process rediploidization. And although the reasons for why this seem to inevitably happen are an active area of research, we think that it's some combination of fixing what dosage problems were created by polyploidization, particularly around sex determination, and the redeployment of what duplicated genes are retained for distinct purposes. And indeed, in an ancestral tetraploid European sturgeon called the sterlet, for which we have a pretty good genome sequence, there's pretty clear evidence that most of the genome has at least rediploidized. But white sturgeon, the subject of our paper, to get to be an ancestral octoploid has probably undergone 2 polyploidization events. The first prior to the split with sterlet, so a shared one, and then one that's more recent and exclusive to Pacific sturgeon. But if sterlet are ancestral tetraploids with functional diploids, what does that make white sturgeon? So what we've gleaned from our genome assembly from white sturgeon is that there's evidence both that the majority of the genome seems to exist in 4 copies, or relative to the sterlet sturgeons, that's a tongue twister, diploid number, which makes white sturgeon technically tetraploid. There's also evidence that 4 ancestral chromosomes don't recombine much during meiosis, suggesting that enough of the genome has rediploidized for the machinery of cell division to treat them as 2 pair rather than 4 of a kind. So notwithstanding that there's ambiguity in our results, because shockingly, it's really hard to assemble an octoploid genome. It looks like the white sturgeon genome is in sort of an intermediate phase of rediploidizing from being functionally tetraploid to the most recent autopolyploidy event. So I guess what I'm trying to say in answering your question is that it's hard and it creates problems, but it seems that most of the time these genomes go back to being diploid across time. Whether polyploidy is an important factor evolutionary is sort of a different question, but in terms of how it sort of rejiggers the machinery, it does that by becoming rediploid, going back to being diploid over time.
So it tries to simplify over time genetically.
Right. Yeah.
In the, what's the word? I guess the background research that I tried to do, and I don't pretend to understand it, they made the case that perhaps this sort of branching ploidy is to deal with evolutionary or environmental conditions. So I guess the argument is plants began that process when they came onto land. Is that it? Kind of where we're going here? The environment changes so much that the genetics had to change really quickly, and one way to do that is to have more chromosomes that can drift and change with the environment.
Yeah, and the broad answer to your question is maybe. I don't want to get into what one might call an adaptationist or group selection paradigm, because not every trait that an organism has is necessarily adaptive. Sometimes it's just neutral, or maybe even disadvantageous, but developmentally connected to a trait that is advantageous. In addition, for any trait to actually be adaptive, it has to be advantageous at an individual level. That is, it has to give that individual an edge over competitors within its own species. So that trait can't just appear to be beneficial for the population as a whole. And the same here goes with ploidy. It's likely that in most cases, these kinds of genomic changes are ultimately lethal. And it's only when the stars align that a polyploidization event happens, that genome is developmentally viable. The phenotype it creates is relatively neutral or even has some advantage in some environment. And then that individual is fertile and able to reproduce either with normal ploidy individuals or that there are enough other polyploids around with him to reproduce. And those are pretty exceptional circumstances. But the answer to the broader question of why would this ever possibly work out is really the same answer to why we have sexual reproduction in the first place, which I think is part of what you're talking about. And the answer to that is novelty. The most salient theory for why we have sexual reproduction, which creates generally diploid offspring using material from two parents, why it evolved in the first place is because of a race between organisms and they're generally clonally reproducing parasites and pathogens. Combining DNA from two individuals creates new variants a lot faster than having to mutate them from scratch. And the same is true for polyploids. When we look across environments, polyploids are associated with two things, as you say, extreme environments and also humans. Humans, and this is mostly true for agricultural species like the wheat that I mentioned, because when we find weirdos that have traits that we like, even if those traits would have put them at a disadvantage out in the wild, we baby them and we breed them a bunch. So like with wheat, most grasses have tiny dinky little seeds, and most of what's adaptive about them is their ability to delay germination, to resist being eaten or resist digestion if they are eaten, or to spread as far as possible. But wheat, on the other hand, has these giant honking grains that we love, but are pretty terrible for avoiding being eaten or dispersed. And it turns out the same goes in extreme environments. A lot of polyploids, where they are viable, have exaggerated characteristics compared to those normal ploidy individuals in their parent species. So think like wheat with the giant seeds. They're often what Stephen Jay Gould once called hopeful monsters, obvious mutants that are just looking for the right environment. And in places where you're competing with normal ploidy individuals in an ancestral environment, you may be at a disadvantage. But if you happen to disperse into an extreme environment, extreme here simply meaning beyond the normal environmental tolerances for that species, immediately you've got less competition from those normal ploidy individuals. And if those exaggerated characteristics make you better suited for that environment than normal ploidy individuals, then you're off to the races. Ecologists would say you're exploiting a new niche. So, but to bring it home to Sturgeon, why sturgeon specifically exhibit this kind of ploidy variation, my guess is that this is genomic flexibility. It has more to do with a slow mutation rate and a compound history of polyploidy than an adaptively retained ability to tolerate or utilize changes. Though, sort of to your broader point, whether that has given them an advantage to weather the many changes that they have seen over 300 million years, that's an open question. Yeah, I don't think we really have the answer to that yet.
Okay, I'm going to go on a short tangent because I don't know if you have the answer. You mentioned extreme environments and environments where you can change quickly, where you have less, other animals and plants being in competition with you. What about things like ocean islands that, volcanic, they're wiped clean? Is this another type of environment where Ploidy can take advantage? So I'm thinking Galapagos, I'm thinking Hawaii, I'm thinking, those sorts of like really extreme but yet isolated environments.
Yeah, so islands have always been sort of a natural laboratory for evolutionary diversification and adaptation. And there are a number of reasons for that. Islands, as you say, they often have less competition because there's so many fewer species, which means that there are a lot of unoccupied niches. They tend to be very isolated, so there's not a lot of input from other populations that would prevent you from adapting to those new environments. And they often have very small population sizes. And so it's technically inbreeding, but essentially what's happening in populations, you always have a balance between things that are happening because of genetic drift. So these are just numerical things that gene frequencies change because there are just a limited number of individuals, and natural selection. Natural selection is creating adaptation. And so those factors are often in conflict with one another, because drift will often sort of accidentally take you away from the adaptive field that you're looking for. I don't want to anthropomorphize it. That is optimal. And so in large populations, natural selection is less opposed by drift, and therefore it can be more effective. And in small populations, drift is a much bigger force. And what that can sometimes mean is you can drift over into different phenotypes that are actually better for niches, unoccupied niches that you wouldn't have been able to exploit before. And that can be true for polyploidy as well. Think of polyploids as being sort of the oddballs that normally they would get selected out. But when you're in these isolated environments where you're not getting input from a lot of normal ploidy individuals, it doesn't take that many polyploid individuals to start an entirely new population. So that, combined with the unoccupied niches, with the isolation, all those kinds of things, gives them an opportunity that they wouldn't normally have in mainland environments.
Cool. All right. So we know they have complex genetics. And I don't really want to get into it, but I will just kind of drop it. We have a society that has gotten confused when talking about the rare cases in human genetics when it comes to sex. But with 4 or 6 different pairs or chromosome groups, like, can they switch sexes like other species? Is there some combination of chromosomes that kind of try to determine this? Like, it's got to be way more complicated for them than it is for us.
It is. It's also in some ways sort of fundamentally different, because, as I alluded to, sex is one of the primary reasons that genomes are forced to restructure after a polyploidization event. So, for example, in mammal species, which have an XY sex chromosome system like humans, females, which of course have 2 X chromosomes, one of them is generally completely turned off during development, or silent, to maintain the same dosage as in males, which only have a single X, right? And an interesting manifestation of this, if you've ever seen a tortoiseshell cat, which have that attractive black and orange spotting, it's because that gene for color is on the X chromosome in cats, and which X chromosome is silenced can vary across the body in early development in females, which leads to different versions of the gene being expressed in different patches of skin. But to your question, part of the reason why it's very different in sturgeons to a degree is because they show a ZW sex determination system. So birds generally have a ZW sex determination system, and that's where the female is the sex with the 2 different sex chromosomes rather than the male. And so, just to be obvious about that, we say ZW instead of XY in these female heterogametic species. And we know that that's true, that sturgeons have a ZW sex determination system because you can actually feminize or masculinize fish using hormones. So when feminized males are used, so this would be a fish that only has the ZZ chromosome, they produce all male offspring because there are no W chromosomes amongst them. Whereas if you masculinize females and make ZW females be male, they produce both male and female offspring, although the ratio is messed up because there are now more W chromosomes than there would usually be. And indeed, in the sterlet sturgeon, that diploid European sturgeon that I may have mentioned before, there is a region on one chromosome that's only present in females, as expected. And it happens to be a gene that's strongly expressed in the sex organs of some other fishes. And then we found the same region present in white sturgeon, too. And it seems to predict sex pretty well, depending on which population you're looking at. But all of that being said, it's true. We don't know how much environment may play a role in determining sex of sturgeon. It is possible that this genomic region that we found, both in these two species of sturgeon, it can be mediated or completely overridden in certain environments. And that could cause some noise around what the markers that we develop from this predict in terms of sex.
Interesting. All that to say is it's possible, but we don't know.
It is possible. So as I thought to say earlier and didn't get a chance, the other wonky thing about sturgeon is that not only are they probably functional tetraploids themselves, they also show spontaneous autopolyploids. So these are the fish with the 6 N chromosomes that you were talking about. And these spontaneous autopolyploids, what we know is that they have 1.5 times the normal amount of DNA normal for white sturgeon, which is, of course, a sort of amorphous concept. But these individuals then 1.5 times would either be hexaploid, the 6 N you mentioned, or if you're talking ancestrally, they could be dodecaploid, 12 N, or if you consider that white sturgeon are normally diploid, they are triploid. And I can talk about how we think that actually happens, the spontaneous autopolyploidy, but the weirder part is, as a rule, triploid fish are sterile, right? In fact, triploid fish, you may have heard of these, rainbow trout, grass carp, Atlantic salmon, are often stocked in the wild specifically because they're sterile, because they won't invade habitats or interbreed with wild fish. And they're sterile because having 3 sets of chromosomes, when they go to make their haploid gametes, the chromosomes get all shuffled up in unpredictable ways, and their gametes end up having uneven sets. That is, they're aneuploid. But I guess nobody told white sturgeon because, while we've found that these 1.5 times variants are rare in the wild, they're still present, but they're rare. They're pretty common in hatcheries, common enough that they've been unintentionally crossed with normal ploidy sturgeon, and lo and behold, they produce viable offspring with an intermediate ploidy, which is probably safest to call pentaploid, I guess, and which is 5 sets of chromosomes. And we don't really know a lot about these pentaploids except that they're viable. So it's sort of an open question as to whether or not they could be important evolutionarily if they were created out in the wild and in the population. But the point, to your point, is that those spontaneous autopolyploids, the 1.5 times fish, that are hexaploid, dodecaploid, triploid, whatever you call them, are at least viable and partly fertile. And you can sort of imagine that there are situations where enough of them were accumulating out there in sort of an isolated environment that they actually could create an entirely new population. And so that sort of gives you a bridge or a window to how these higher ploidies could have formed in the first place. And you have these sort of strange cellular developmental things going on that result from sort of breakdowns in environmental conditions, and just create this sort of convergence of conditions where a new set of ploidy variants happen to start reproducing and diverge from the original population.
Okay, so let's go back to the paper. You're using DNA markers to figure out whether or not individuals are male or female. And according to this, you can't see, I would imagine, a fish, whether it's male or female, just by looking at it. And so you're using genetics and you're 96% accurate for females, but only 81% for males. Why is it easier to spot the female genetically? And is it a null result sometimes instead of, like, say, a false positive or a false negative?
Yeah, it's certainly possible that some cases where our sex markers aren't 100% accurate could be because of the failure of the molecular assay, something that we call a null allele, where the genomic region is there in that individual, but our assay simply fails to pick it up. And that would explain why some females were predicted to be males, but not why males would be predicted to be females. In those cases, it's not the assay that's failing, since we can clearly see that the genomic region is present, but rather a failed association of the genomic region with phenotypic sex. And I think what's actually going on is any or all of three things. One, it's possible that the sex data for our samples are wrong for some populations. That is, which are males and which are females was just misrecorded. As a scientist, you have to be upfront about the fact that your data may just be flawed in some cases. And the fact that our markers are 100% accurate for fish whose sex I have personally confirmed, not by looking under the hood, but through offspring of known crosses, sort of indicates that in some cases. But two, as we said earlier, it's possible that in some cases, environment may play more of a role in determining sex than we understand. But three, and this is my preferred hypothesis, or at least the one I'm most interested in pursuing, that how far down the process of rediploidization, and specifically how much exchange between those ancestral sex chromosomes there still is, varies from one population to another. And so the association of our markers, and perhaps even how that genomic region mediates sex, may vary from one sturgeon population to another. And indeed, some of the data that we use to make our genome assembly indicate that at least in the Middle Snake River in Idaho, which is one of the places where our sex markers aren't 100% accurate, there is still some recombination between those ancestral sex chromosomes.
Okay, well, let's try to give this a little bit more real world heft to it now. How does this, I guess I'm going to add something to my original question, is like, how does this further our understanding of genetics in the animal kingdom, and how do we use this to, like, help conservation of this species? Like, how do we use this information in practice?
Yeah, it's been a few decades now since we sequenced the human genome, and we know a lot more than we did then. But every day we sort of find out that our understanding of what genomes are, how they work, what's wrong, and that's science. That's how that's supposed to work. So for example, these days we're interested in structural variation. So structural variation of genomes is entire sections of the genome that are present in one individual or missing in another, or regions that are reversed so that they're in the opposite direction from one individual to another. We sort of originally thought that genomes were basically the same, except I've got an A and you've got a T, and over here you've got a G and I've got a C. That there's very small differences. It turns out that even among individuals in the same species, there can be huge, very big differences in the chromosomes between individuals, and so that is particularly true for polyploids, where not only is that happening between just very small homologous chromosomes, but chromosomes that have a much larger ancestral relationship. And so the way the genomes evolve or rediploidize following polyploidization provides a good window into understanding that. But as to the conservation of this species, which of course is what I'm interested in on behalf of the tribe, it's easy to imagine that folks running commercial caviar farms would like to know which fish are female as early as possible so they can maximize their return on investment. But that's not really our target audience. On behalf of the tribes, we're focused on trying to maintain or rebuild the fisheries of these species, and understanding population dynamics is a critical part of that, like the sex ratio of wild populations, or how many females reproduce in a year and with how many males. And being able to take a thin clip and predict sex rather than having to do an abdominal biopsy makes that data a lot more accessible. But beyond that, because there are a lot of populations that are slowly declining because of a lack of consistent recruitment and reproduction, a number of conservation aquaculture programs. So these are hatcheries that are specifically focused on rebuilding wild populations have been developed to supplement those wild populations. And while they have become very adept at producing many young sturgeon, they're very effective. We don't know whether those aquaculture practices bias the resulting offspring towards one sex or another. For example, it's possible that male sturgeon grow faster than females. And at high densities, like there are in hatcheries, males may outcompete females and produce, therefore, a male biased population. You don't want to dump a whole bunch of males back into the wild because that screws up population dynamics. So since you can't sex young sturgeon until they're basically begun to mature, having a molecular assay like we've developed from this marker is a much more effective way of doing that at earlier life stages.
Okay, so you're providing the information needed for those that are making the policy decisions about how to do the conservation. You're giving them the tools to make those decisions.
Yeah, we're much more focused on sort of the state agencies that are putting into action. Policy is something that we leave to the people who have, in a higher pay grade.
No, right, but you're giving them the information to make those policy decisions. I mean, in a volcanology sense, a volcanologist isn't the one telling people to evacuate. The volcanologist is telling them, okay, this is what's happening, this is the likely outcome. And then the policy people who make a lot more money typically, not always, but typically make the decision whether or not you need to get out of town.
Yes, exactly. We provide as much information as we can, and what they choose to do with it is beyond our purview, unfortunately.
Yeah, exactly. Okay, so I'm kind of glad to see that it's working similarly in two completely different disciplines, actually.
That's science, yes.
For better or worse, it goes both ways. Sometimes you really don't want the scientist making the decision for people sometimes, and sometimes it would actually be better, but I'm biased there, I guess.
Yeah, no, having everybody at the table is certainly the right way to do it. It's just being able to respect everyone's opinions. It's sometimes a challenge.
Yeah. On that note, I'm going to skip one of my questions because I think it's kind of wrapped up here in the same. So the Fraser River, it's a river that connects my home turf, and more broadly for my local river. What is the future for this species? Because you've mentioned the Snake River already being already quite different, but I know you're focused more on the Columbia and its tributaries, but what can you say about the Fraser and what needs to happen to protect it?
Yeah. So as we said, the Columbia has a major challenge of having all of these dams. And you can imagine that prehistorically, a 5-meter fish could go basically anywhere it wanted in the basin. If there are different sections of the river basin that are better for reproduction, for growth, for, hanging out in hot summer temperatures, those kind of things, you could choose that. And you put in dams in the way, well, now you just got to deal with whatever is available there. And unfortunately, in many of these sections, there's not enough good habitat for rearing of the early life stages that they're getting a lot of recruitment. And so what is there is getting older and older, and as a result, the populations diminish over time. The Fraser does not have that problem of damming. From my understanding, the biggest threats to the Fraser white sturgeon are habitat degradation and the diminishing runs of anadromous fish, particularly chum, salmon, and eupilon. It may be strange to think of a freshwater fish as being dependent on marine species, but these interior Pacific ecosystems have evolved with that regular influx of marine-derived nutrients. And that's especially true for white sturgeon, which depend on those returning fish, their carcasses, their eggs, their offspring for food. And those runs diminish. As those runs diminish, it's likely that sturgeon will continue to do so too, unfortunately. And we're already hearing reports of, and I should say that's not something that conservation aquaculture can overcome, because you can dump a bunch of young fish in the river, but if there's no food for them to eat, then you're just spinning your wheels. And we're already hearing reports this year of another warm water blob developing off of California, and the prediction of a super El Nino later this year. So it seems to be that while there are things that we can do to give the Fraser River or the Columbia River sturgeon the best chance we can, their fate, like the fate of so many other species, is going to depend on whether we humans can get our act together as a species, realize that we're not entitled to be the center of the universe, and start acting like good stewards like we should be.
So we could sum it up with essentially climate change.
Yeah. I mean, that's certainly the train coming down the tracks, right? It's how long do
Yeah.
we need the train before we decide that we're going to get off the tracks?
Uh, and I'm actually more of an optimist than a doomsayer because I think being a doomsayer paralyzes action. We are doing stuff. Things are changing. It's just changing too slowly, to be fair.
Yeah.
But
Well, I mean, the science is there, and it's really just a matter of political will when it comes down to it, which is both promising and depressing.
yeah, it's both simultaneously,
Yep.
100%. Yeah, it's one of these days. Actually, as a plug, I don't know if it'll be my last episode of the season, but because of just the nature of when things need to be published, because I have another one that I have to record that I don't know when I can get out. It's going to be all about volcanoes and climate change and how we know that volcanoes do not put out nearly as much CO2 as humans. I'm going to go through the nerdy math and the melt inclusions and the whole 9 yards. So that I don't know if it'll help, but you know what I mean.
Yeah. No, dispelling any alternative than we're doing it to ourselves is definitely part of the puzzle.
Okay, so we're nearing the end and it's time for callouts. This is your chance to flag cool projects, something you're working on, something completely random. It's whatever you want.
Yeah. I mean, really, I just want to acknowledge all the people that have contributed to this. What we do with Sturgeon is a team effort, and we have a number of irons in the fire. We're looking at spontaneous autopolyploidy in wild fish because it seems to be happening in the wild as well in really hot weather years. We're continuing to look at how, things like shad, which are the big thing in the Columbia River Basin now, huge runs of shad that turn out to have a lot of something called thiomanase in their tissues. And that thiomanase causes the eggs to be thiamine deficient and for these fish to die or grow weird. So we have a number of projects that we're working on, and all of them depend on our collaborations with state agencies and universities to work. And in particular, I want to acknowledge the leadership of Janella Miller, who's now a manager with Critvic, but was formerly with the Yakima Nation Fisheries and developed their conservation aquaculture program. And she has been just a huge, steadfast champion for understanding and rebuilding white sturgeon populations. And I'm really glad to be able to work with her.
Awesome. You know what that means? It's time for the infamous question. The one that I warn every single guest is coming. I warn it before they even see what kind of topics I'm going to ask them or anything else. It is the traditional last question. What is your favorite science joke?
Yeah. I don't know if I would call it a joke or a lament, but I have always enjoyed the description of the kind of work that I do by a guy named John Shepard, who's a sort of a famous fisheries professor. He said that studying fish populations is exactly like studying forests, except you can't see them and they move. Which I guess makes white sturgeon like the giant sequoias of the freshwater fish world. So I'm glad to be working on that.
Perfect. Well, thank you so much for taking the time to come on and teaching me a lot about white sturgeon.
Yep, I'm always happy to talk about sturgeon and genetics, my favorite things.
And that brings the second to last episode of Season 2 to an end. Okay, so that means that next episode is the last of Season 2, and it's going to be a solo episode, all about volcanoes and CO2 and climate change. A little bit of a historical romp, I guess, through how we can prove that volcanoes do not put out anywhere near the amount of CO2 that humans do. A fitting way to end Season 2. At any rate, just remember that over the summer there will be encore editions to kind of fill the feed to make sure that the computer overlords are still happy with whimsical wavelengths. And then we'll be back in September. There are a few things that could get into the mix, and I'll repeat these next time, because I'm also expecting a surgery somewhere over summer, and I really don't know when that'll happen. So hopefully everything lines up, and I'll be able to start Season 3 on time, September 14th. All right. I'll see you again in 2 weeks.
In a world so chaotic, so misbehaved. Echoes of melodies remind us it's all waves. Colors weave stories painting the sky, swaying to rhythms as the galaxies fly by. Her